Go ahead and get started. So hello, everyone. If you've already had a nice break and ready for the new year, it's my pleasure today to introduce Dr. Jun Cai. And also thank you for coming to the Center for Integrative Environmental Health Sciences and Department of Pharmacology and Toxicology. Welcome you to the Environmental Health Science Series seminar. So Dr. Cai currently holds an associate professorship at the Pediatric Research Institute of the Department of Pediatrics. He received his MD at the Tianjin Medical College and his PhD in biochemistry and molecular biology at the Tianjin Medical University in China. He then continued to pursue his postdoc training in developmental neuroscience, especially in the field of oligodendrocyte development that was supported by the National Multiple Sclerosis Society at U of L. Dr. Cai is a productive scientist in the fields of neural development and neurotrauma. His research has largely been supported by the NIH and foundations. He served as a peer reviewer for over 40 academic journals and ad hoc grant reviewer for the federal, national, and international granting agencies. He's also served on the scientific review committee for the Sleep Research Society and the Fellows Scholarship Oversight Committee of the American Board of Pediatrics. Dr. Cai is a member of the Society for Neuroscience, National Neurotrauma Society, Society for Experimental Biology and Medicine, Sleep Research Society, and holds a full membership of Sigma Xi, I think I said that right? Sigma Xi, the Scientific Research Honor Society. His research focuses on molecular and genetic regulations of white matter development in the CNS and the mechanisms underlying CNS impairments and regeneration in diseases. He has published over 90 original research papers in peer-reviewed journals, including Neuron, Journal of Neuroscience, GLIA, Journal of Neuroinflammation, Development, Stem Cells, and the Journal of Pathology. So today he will be giving us talk titled Roles of Early Life Cadmium Exposure and High-Fat Diet in Neurodevelopment Disorders. Ready?
Well, thank you, Tyler, for the introduction. I came to Louisville in 1998, so that's got my postdoc training at the anatomy department. So during the next 20 years, my study focused on the oligodendrocyte development. That's kind of a myelin-forming cells in the central nervous system. And I remember that at the end of 2020, and Lou pushed me to submit a pilot grant to the center. So since then, I stepped in a totally new field for me. So today I'm going to share the story just based on this pilot grant supported project. So the title here, the roles of early life cadmium exposure and high-fat diet in neurodevelopmental disorders. But I think this is most popular slides on the internet and tell us the cell types in the nervous system. In adult human brain, there are about 100 billion cells, including the 10% of neurons and 90% of glial cells. So in the glial cells, there are two cell types. One we call the macroglia, and the other one is microglia. So astrocytes, oligodendrocytes, and ependymal cells are macroglias. But in the peripheral nervous system, they don't have the astrocytes and oligodendrocytes. Instead, they have satellites and Schwann cells. All of these cells execute a similar function as astrocytes and oligodendrocytes in the central nervous system. So all of these cells, what they come from. So at very early stage, early embryonic stage, there's a small molecules we call the sunny hedgehog is synthesized and released from node cord and induce the neural tube, the neural plate folded to form the neural tube. And the most inner layer of the neural tube we call the neuroepithelium. And this epithelium is eventually formed the different progenitor domains. controlled by the different transcription factors. So these transcription factors is induced by the both dorsal midline signaling, you call the BMP, and ventral signaling, midline signaling, sunny hedgehog. So they interact each other, and eventually they define the boundary of different progenitor domains. So here is, we can see that, for example, in the ventral part of the neural tube, we can see here there are 5 progenitor domains, including the motor neuron domains. So from motor neuron domain, the first, it generates the neuron progenitor cells, and then followed by the oligodendrogenesis. So in other domains here, they first generate the interneuron progenitors, and then generate astrocytes. So this is where those neural cells in the central nervous system, where they come from. And so these cells, they generate from ventricular zone, and then they leave the ventricular zone, migrate to the destination area, and start to differentiate to become the mature cells over there. So after they move to migrate to the destination area, and there's several very important neurodevelopmental processes happened. And so here we compare the human and the mouse. We can see that in the human, the neurogenesis occurred pretty early at the first trimester, and the gliogenesis and the synaptogenesis happened start from the second trimester, and the synapse forming is just start after the birth, and all of these events roughly finished around 20 years old. In mice, there's a little bit difference. The neurogenesis and synaptogenesis is just start before the birth, and myelination, this event is just after the birth. So here, just tell you that all of these neurodevelopmental important neurodevelopmental processes has happened from the beginning at embryonic stage, and they even go further to the adulthood. So the maternal disease and the so that's we can easily understand that all of the mutations, the gene mutations, maternal disease, and the environmental factors will affect the neurodevelopmental outcomes. And so the maternal disease and environmental factors, including that the obesity, the mother obesity, gestational diabetes, viral infection here, air pollution, smoking, and also the social economic status and stress, depression, and autoimmune disease. All of these activate the maternal immune system response. And these activation increase pro-inflammatory cytokines such as TNF-alpha, interleukin-1 beta, and the interleukin-6. And all of these molecules in inflammatory cytokines and go to the placenta and active the placenta immune response. And these are associated in the placenta immune activation and also the inflammation. So all of these associated in the cytokines release immune cell activation, oxidative stress, and even change some neurotransmitter signaling, such as the serotonin. And these molecules go to the fetal circulation and reach the fetal brain. And these also activate the fetal brain immune response. So the fetal brain response to all of these molecules are associated with oxidative stress, microglia priming, impaired synaptosis, and also change the neuron migrations. So the neuron migration, those neuron cells, they probably move to other areas. It's not their target area. And eventually, and even just disturb the axon targeting event. So this eventually result in the offspring developmental outcomes. So this it lead to the high risk to develop the neurodevelopmental disorders. So here we can see that the disorders including autism spectral disorders, attention, ADHD, and intellectual disability, communication disorders, and also include other mental disorders. Here we mentioned that schizophrenia spectrum and depressive and anxiety disorders. So we know that there's a lot of genetic factors, both genetic and environmental factors, impact on the prenatal and the postnatal area on the brain development. So here is most recently is a cohort study from clinical side in Norway, and they analyzed a lot of data, and eventually they recruit almost 15,000 mothers and 15,000 fathers and look at their offsprings. So these studies show that the maternal factors such as the maternal age, cigarette smoking, alcohol consumption, and also the depression and the anxiety symptoms, they are the high risk for the neurodevelopmental disorders. So here they listed the ADHD, autism, and also the schizophrenia. This is another cohort study conducted in Korea, and they investigated prenatal exposure to the father smoking situations. They recruit almost 16,000 target population in 13 cities in Korea, and then they replicate the samples only in one city, almost 50,000 target population. After the first initial screen, eventually they target over 1,000, over 10,000 in discovery sample and almost 30,000 in the replication samples. They found that the paternal factors can substantially contribute the ASD risk for their offspring, especially in the case that the father smoking both before and during pregnancy contributes significantly ASD risk. So it's very reasonable. We look at what happened in the smoking during the pregnancy, and we know that when we smoke a cigarette, it's generate over 4,000 chemical compounds, and we've already know that the nicotine is play a very harmful factors on neurodevelopmental disorders. How about others? So here we can see that when you're light up, it generates the three major heavy metals, the lead, cadmium, and arsenic. So all of these three heavy metals also are also found in air, soil, and water. most recently, in 168 baby food are analyzed, are tested, and find all the lead, cadmium, arsenic, and mercury is just exceeds the minimal requirement approved by FDA. So this is raised a huge health issues for the heavy metal contaminations. And we know all of these, we listed all of these heavy metals. They have two common consequences. One is they can activate immune systems, and second, they impact on the brain function. So this is here, it's a reasonable question. Is early life exposure to heavy metals associated with neurodevelopmental disorder? So Dr. Han and her colleagues revealed the epidemiological data from 1970s to 2021, and they collect 17,000. They collect 17,000 reports from the publications, and eventually they find the 100 publications reach their criteria. So this is come from those 100 papers, and they find that the childhood lead exposure and increase the risk of neurodevelopmental delays and behavior problems. And at least, all of those have one domain of the neurodevelopmental problem. And the lower prenatal exposure to lead, and even the blood lead level is less than 5 microgram per deciliter, has showed adverse effect on neurodevelopmental in infancy and early childhood. So arsenic, the postnatal and maternal arsenic exposure cause the deficits in cognition and the motor domains. Among these, and the cadmium is the only heavy metal they found from clinical side. They found that associated with social ability. So there's no doubt that that's why the National Toxicology Program, they confirmed that the adverse effect of the lead on cognition, enhanced attention-related and other problem behavior. So there's no problem for his. But for the arsenic and that. Okay, so the arsenic and cadmium impact, we need more investigations. So early life cadmium exposure, I think rather than the lead and the arsenic may contribute significantly to the ASD risk due to the because of the social deficits. So I think, well, this is for me, I think for your guys, it's some basic knowledge. And cadmium is the 7th most toxic metal, and distributed in many metal ores, and also found in air, water, soil, plants, and house dust. When the human use that, the phosphate fertilizers and fossil fuel burning, and battery manufacturers, jewelry and paints and smoking, and all of these generate the cadmium contamination. So the human exposures usually came from two major routes. One is from diet and drinking. The other one is from the breathing, such as the air pollution and smoking. And also in some workplaces like mining, battery manufacturing, soldering and welding. They also generate the cadmium cadmium contaminations. So when the cadmium get into the body, so if those cadmium came from food and drink, they absorbed it by the GI tract and get into the circulation. They can combine with the albumin and the MT proteins and GSH. But most of the cadmia, they transport it to the liver and stimulate the liver to synthesize the one metal binding protein. We call that the metallothelins. MT proteins easier for me. Okay, and very similarly, and if the smoking or the cadmium from the air or smoking, and they get into the lung cells also synthesize MT proteins. This is a very strong binding proteins and bind with cadmium. And all of these MT bound cadmium and go back to the circulation and delivered to the kidney. And then we can see that in the kidney, if the kidney get injured, we can see all of these proteins, find these proteins in urine. So most of the cadmium in the body, they stored in 30% of the cadmium stored in the liver and 30% in the kidney. So these two organs is the most organs containing high level of cadmium, and also the clearance of cadmium. The clearance half-life is around 20 to 30 years, and it's pretty slow. The body can get rid of those cadmium by the urinary and the fecal routes. So all the cadmium can accumulate in the body for a dozen years and may induce some damage for the organs. And also the cadmium can freely pass through the placenta barrier and exist in the breast milk, and also can go through the blood-brain barrier. And this table on your left hand, this is the safe level. The table is summarized the safe level of the cadmium. It's defined by WHO, EPA, CDC, and FDA. So we can see the bottled water is required to less than 3 microgram per liter, defined by WHO, and in the United States, it's less than 5 microgram per liter. And I think this, and also the water, the reference dose for the water is less 0.5 microgram kilogram body weight per day. So here, the blood cadmium level is supposed, the safe level is less than 0.04 microgram per demiliter. So that's, we just want to test, based on those information, we want to test our hypothesis on the animal models. And so this animal model is, it's widely used on campus. And the mice, the female and the male mice came in and get used to the environmental for 1 week, and then we start to mate them and feed them with drinking water containing either 0.5 or 5 ppm cadmium, and then the F1 mice were continually fed these drinking water contain the cadmium until the terminations. So we define these ppm, the cadmium level. I think is based on the human cadmium reference dose in water. So if we count like the 30 gram per mouse. So each mouse, they drink 7.8 mils per day. And so the totally, suppose the mouse take in the cadmium range is between the 3.9 to 39 microgram, 39 microgram for each mice each day. And the diet, we use the 10% fat, the normal diet. So it's a reasonable question. We need to validate the early life cadmium exposure mouse model. And so the first, we review the data from both human and the animal model published before. We try to find the animal model most close to our model. So this is it's not too much. So here is what we find from human data, and most of the studies just focus on the kidney injury, and they found that when they exposed to the cadmium and the blood cadmium level is just over the 0.1 microgram per deciliter, and they find some protein urine. And also the MAG alpha-1 microglobin, beta-2 microglobin, and kidney injury molecule, and albumin and lysozyme. All of these molecules increase in urine and indicate that the tubular toxicity. We only find one animal models, but they applied by 3 different labs, and they use the 8-week-old mice and they exposed to cadmium chlorides in drinking water. So this is at the dose of 10 microgram per liter. It's very close to our 5 ppm dosage. The 5 ppm dosage is just over the 8 microgram per liters. So they expose those mice for 16 to 23 weeks. And they studied on the kidney injuries and also the liver injuries. They only find, and even exposed to the 23 weeks, they only find some vacuole and lysosome increased in the tubular cells, but they never find that the proteins in the urine. And in liver study, they find ALT, AST, triglyceride, oil O staining, and MT protein were found significant increase in the liver tissue. So this is we get from the literature. And the good things, we have the detail about the liver injury, use this model, and it's published by Dr. Yang in 2022. And we can look at here, I just extract the data only exposed to the cadmium at 5.5 and 5 ppm. And we did not see any difference in the body weight. And those mice exposed to cadmium show the normal glucose and the insulin metabolism. So for the liver function, we find the ALT level is significantly increased in the mice exposed to 5 ppm, and also the AST have the trend increased in those 5 ppm cadmium exposed mice. And all of the liver morphologies looks good. And of course, the liver MT proteins is significant increase only exposed to 5 ppm at both transcriptional and protein level. So from this data, and we can see here the early life exposure to the low dose cadmium at 5 ppm causes mild changes in offspring mouse liver. So here we just compare our data to the previous publication on the mice study. So we can see here, I hope that we can get the kidney data from either the Jamie or from Madhuri. I didn't get those information yet, but from the liver injury, and we compare that in our study, we only find the ALT, AST, and MT significant increase in the liver tissue. And we didn't see the morphology pathological changes. So we concluded that the early life exposure to 5 ppm cadmium in drinking water may cause a marginal risk for liver and the kidney. It's question mark. I still need those data. So we examined the cadmium levels in brain. So we dissect 3 different areas in brain, the cortex, hippocampus, and cerebellum. We found that only when the mice exposed to 5 ppm cadmium, we can see the significant increase the cadmium level in cortex, hippocampus, and cerebellum. So when we talk about, when we talk about the neurodevelopmental disorder related behaviors, we first run the bunch of animal behavior tests. So here we choose 3-chamber test. We'll evaluate the social ability and the social novelty, and the Marble-Berry and self-grooming test evaluate repetitive and restrictive behaviors. All of these 2 behaviors are core symptoms of ASD, autism spectrum disorders. And we also examined other behaviors. We use the Y-maze to test the spatial memory, open field for locomotor activities and anxiety-like behavior, and elevated plus maze. This test is for the anxiety-like behaviors. So all of these have some symptoms occurred in the ASD and also other mental disorders. So here is the 3-chamber test, and we have 4 sessions, and every session have 10 minutes, and we just put the tested mice in the middle chambers and close on the both sides, close the door, and let the mice freely run in the center chamber for 10 minutes, and then we open the both side, the door, the both side on the isolating board. And so the mice can travel on both sides and get used to the environmental and also this cage. And then we put the third sessions, we put 1 mouse and enter the cage. And the mice, the tested mice, usually they like to contact with these strangers and spend more time with it. And so they ignore that the other side. And then we put a novel mouse in under another cage. So the mice, the normal mice, they're supposed to spend more time with these novel mice, and they just spend less time for those familiar the mice. So this is the whole process. And we can look at here the 13 weeks. The 13-week-old F1 males, we only test the males here, and we didn't find the significant difference in social ability between the control and cadmium exposed mice. And compared to the control mice, the 13-week-old male mice exposed to cadmium show a declining trend of social novelty, but it's after another 14 weeks, and surprisingly we find in these 27-week-old male mice exposed to 5 ppm cadmium had a higher social novelty than those exposed to the 0.5 ppm cadmium. So this is just, yeah, surprise us, the result. And then we run the marbleberry and self-grooming. And the marbleberry, we just put 20 green glass balls on the surface of a corn cob bedding. And if the mouse buried the ball over 2/3 of the volume, and we consider that's positive. So we take recording for 30 minutes. And here is the result. And we compare to the control mice. The mice exposed to cadmium did not show a significant difference in marbleberry. There's no difference. And this is a grooming test. So we just let the mouse in the cage for 5 minutes, get used to the environment, the cage environment. Then we take record for 10 minutes and calculate how many seconds they spend. They just have the grooming behavior. So we feel so happy for these results because we eventually find the positive, like what we expected. We find that the mice exposed to 5 ppm cadmium, they show they spend more time for grooming compared to the control and the mice exposed 0.5 ppm cadmium. And these start earlier, like 30 weeks old mice, and continue to 27-week-old male mice. So the this is a Y-maze. There are two kinds of Y-maze tests. One is we used for test spontaneous alternation related on a prefrontal cortex function. So usually the mice in one arm, and they have the innate tendency, they explore the novel environment. So they move out from this arm and get the new one. And then when they get out of this one, they try to the third new one. But when the prefrontal cortex is damaged, and they probably these sequences totally disrupted, they may go back to the old one. And the second kind of Y-maze we call the delayed version of Y-maze. This is based on the hippocampus function and evaluate the spatial memory. And so the first time we just put these mice get into the Y-maze, we block one arm. That arm considered as a novel arm, and let the mice run freely in these two arms. And then after 5 minutes, we take the mouse out and we wait another 2 hours or 24 hours. That's we call it the short-term and long-term spatial memory. And then we put the mouse back and we open the novel arm, the block for the novel arm, and then we run another 5 minutes. We are going to calculate how many times the mouse spent in these novel arms. So for the normal mouse, usually they spend more times in that novel arm because that's the new environment and attract them. So we compare these 30 weeks old males and 27 week old males, and the mice exposed to cadmium show a significant decrease in arm entries. So it seems like those mouse, they don't like to move, and or the frequency is pretty low. And we didn't see any alteration of the time in novel arm. We didn't see the difference for those two reference. And so it means that at least based on the Y-maze analysis, we didn't find either the spatial working memory deficits or the short-term spatial memory. Then we go for the open field. So the open field is pretty easy. We just put the mouse in these square box and recording for 10 minutes. And we analyze the first thing, the travel distance. So consistent to the Y maze. And we can see here the mice exposed to cadmium, either the 0.5 or 5 ppm cadmium. And they show they traveled much less distance compared to the control mice. These phenotype is come from the 13-week-old males into the 27-week-old males. And for the evaluated plasmids, this study is we just leave the mice on these maze for 5 minutes, and usually the normal mouse stay in the closed arm for longer. It's about 60% of the total time they spend, and they stay in the closed arm, and 40% in the open arm. So we didn't see any difference, but the mice exposed to 0.5 ppm cadmium spend more time in open arm. This is a trend, but cannot reach the statistics significance. So here is a summary, and we listed the core symptoms of autism, and this is the behavior test, what we used, and exposure to cadmium. So from this table, eventually we can say that the early life low dose cadmium exposure may cause 4 symptoms of autism spectral disorders, and also induce a low local motor activity. So now we talk another important environmental factors affect the youth health, obesity and the overnight. So from here we can see that obesity has more than tripled since 1970s, and right now it's every 5 children have 1 children or has the overweight or obese. So this is a big issue in the United States. And prevalence of obesity remained very stable increase. It's always above the 70%, and currently affect about over 12 million children and adolescents. So at different age range, so we can see here when the children grow up, they have more of them get overweight or obesity develop, and 80% of obese adolescents will become obese adult. There's a lot of reasons can cause obesity, such as the lifestyle, no exercise, and pregnancy can induce the obesity, and also the emotional or psychological factors, kind of medical conditions, lack of sleep. All of these together, they just can contribute for the risk to develop obesity. So obesity is very similar like autism disorders. They have we need to consider 3 different factors from environmental, genetics, and behavior. And overweight and obesity result from basically result from the energy imbalance. So all of these cause a lot of problems, such as depression, social isolation, and sexual problems, and disability. and cause kind of disease like heart disease, stroke, type 2 diabetes, metabolic syndrome, as well as the reproductive problems. So this is a big health issue, not only for the for adults, it's also for the youth, for the kids. So is the obesity and overweight associated with neurodevelopmental disorders? So we just reveal the data, and we find that in the ASD, if you look at that here in the ASD, along the age, we can see the obesity and the overweight is increased when the age increased. And the similar results also observed in the ADHD. And the clinical study also support association between obesity and the neurodevelopmental disorder like the ID, ADHD, and the ASD. The imaging research show that most of obesity and overweight kids, they have the white matter injury impairments. They show a lot of white matter area, is especially for the corpus callosum area. They show the hypomyelination, and they have the fine neural fibers, the axons. So that impact a bunch of function, the neural function. And for the animal study, the high-fi diet can trigger the hyperactivities, anxiety-like behavior, ASD-like behaviors, and the learning memory problem in the wild-type mice. And also the high-fi diet can enhance the ASD-like behaviors in the BDBR mouse model of ASD. So this is from both clinical and the animal study. So we just expose the mice to the high-fi diet. This project is come earlier than the Cadmium project. We start 4 years ago. So I combined some results together. So I don't want to talk about this. This is a typical, the body weight. You can see a significant increase if we expose the mice to high-fi diet. And the glucose, the blood glucose is increased, but never exceed 250 microgram per deciliters. That means they never reach the level of hyperglycemia. And other things is like the IPGTT and the plasma insulin and other things. It's just all of these blood profiles is showed that the high-fat diet feeding for 24 weeks is reliable and can generate the major body weight and blood profiles of obesity. So we repeat those behavior, and we didn't find the significant change for the social ability and the social novelty. And Mubbleberry and self-grooming didn't show the difference. The Y-maze. The Y-maze, we only find that 27 weeks old obesity male mice show a significant decrease in arm entries compared to the control mice. So this is very similar to our Cadmium result. It's only things difference for the open field test, and we can see here the mice exposed to 5 ppm. No, sorry, and the mice exposed to the high-fat diet, and they spend more time, much less time in the center of the open field. So combined with they travel much less distance in the box, and these two put these two results together, it suggests that the mouse exposed to high-fat diet has kind of anxiety-like behavior. But the typical test for elevated plus for the anxiety-like behavior use that EPM, it didn't show that result to support our open field findings. But interesting that if we look at this, the time the mouse spent in open R, and we find that they have the trend to increase their time stay in the central zone. So we just keep this in mind, and we can look at that. So here is a summary for behaviors in mice experience the high-fi diet exposure, and we didn't see social stability, social novelty, and only here. Let me see. Yeah, we have talked about this. The travel distance decrease and time in center decrease. All of these, the high-fi diet induced obesity may lead to anxiety-like behavior, but not core symptoms of autism spectrum disorders. Now we put these two factors together. So this is the liver data and reported by Dr. Young, and I don't want to talk about this here. So we measure that when we put these two factors together, and we measure the cadmium levels in the brain in different areas, and we didn't find that the high-fi diet can either inhibit or increase the cadmium accumulation in the brain tissue. It's the result is there's no difference between the cadmium exposed to 5 ppm or combined with high-fi exposures. So this is the result. We conduct the two-way novel analysis, and I just go through this, and we just see the summary. So I just listed the findings from behavior side by side, exposed to cadmium, exposed to high-fi diet, and put these two factors together. So here we can see that the cadmium exposure is playing more roles on the social communication, like the social novelty and self-grooming. And the high-fi diet play majority roles in the anxiety-like behaviors. So put this together, we conclude that the early life low dose cadmium exposure rather than high-fi diet may cause core symptoms of autism spectrum disorders. Activities. So this is the result is very consistent. Okay, so then we look at the pathological changes under this behavior. And no matter it's cadmium or the high-fi diet, generally we know that all of these two factors, they are going to induce the oxidative stress and generate the RS, and eventually they cause the organ toxicity. This is a typical the concept right now accepted in the field. And also some studies show that cadmium can induce the damage of the BBB, the blood-brain barrier integrity. But controversial results find from high-fi diet treated mice. And some data show that they can impair the blood-brain barrier, but other studies show and even show that they have the protection effect on the blood-brain barrier. So the first, we would like to see the oxidative stress, and this result is published by the group on campus. That's Dr. Friedman from his group, and they find that the 5 ppm cadmium exposure can enhance the high-fi diet induced the ROS and the oxidative stress in 3 different areas, the cortex, hippocampus, and amygdala. So we look at that other oxidation marker, the DNA marker, and we look at the several different areas, prefrontal cortex, and dentate gyros and cerebellums. So it's pretty clear. We can see that when the mouse exposed to 5 ppm cadmium, we can see more this red positive, DNA positive staining show here. And when we have the high-fi diet factor together, and we look at more severe DNA oxidations here. And similar result we can look at, we can see from the cortex and the DG area. And this the single high-fi factor is not that much induce the DNA oxidations in prefrontal cortex area and cortex. So this is more details for the hippocampus area. We look at the CA1 area, CA3 area, and subculum area. So these areas. So we can see the high-fi diet enhance the cadmium-induced DNA oxidation in these areas, especially for the CA1 to CA3 and this subculum. 0.5, we didn't see the difference here. So we also look at the BBB integration using the albumin, because when the albumin leak in the brain, usually the albumin cannot go through the blood-brain barrier. But when the blood-brain barrier impaired, and more albumin leaked in the brain tissues. So here we can see the 5 ppm, we can see more albumin and the grain staining can find in the cortex area. And this is both the cadmium and high-fi exposure. We can see the more staining there. So the blood-brain barrier here in our model is disrupted in offspring cortex exposed to the cadmium at 5 ppm dose, and more severe BBB disruption occurs in the offspring exposed both the cadmium and high-fi diet. Then we look at the glia activation. We check the exercise and microglia. The APOL1 is a microglia marker, and the GFP is an exercise marker. We can consistent with the oxidative stress data, and we can see here that both high-fi diet and cadmium can activate microglia, the microglia, and exercise in cortex and the DG area, but we didn't show the data here, but not in the cerebellum. Because of those inflammatory glial cell activated, so we also check the cytokines in cortex exposed to the cadmium and high-fi diet. We only find that TNF alpha significantly increased when the mice exposed to cadmium, 5 ppm cadmium, and also the interleukin 10, the similar result increased. We checked the TGF beta-1, interferon gamma, and interleukin-1 beta and interleukin-6. We didn't find the difference. So no matter what, put this together, it suggests that the mice exposed to 5 ppm and also the high-fi diet, they can induce the inflammation and inflammatory response in cortex and hippocampus area. Then we would like to see, of course, there's a lot of work to do for the pathological analysis. Currently we try to expand those data, but at the same time we would like to see what kind of signaling pathway underlying these pathological changes, and we focus on 2 signaling pathway. We just target that because the mTOR and Wnt beta catenin signaling pathway is find that it's play a very important and a key role in the autism spectrum disorders. So here is some clinical data and show that the mutation in various components involved in the mTOR signaling and the Wnt signaling. And no matter it is up-regulated or down-regulated, and they can cause the ASD-like pathological changes and behaviors. These data come from both supported from both clinical and animal studies. The mTOR signaling data is come from earlier, and we find that the mTOR signaling is downregulated in the obese mouse brain, but upregulated in mouse, but upregulated in the obese adipose, liver, and heart, and heart and the kidney. So it seems like the brain. Oh, okay. Okay. So this is Wnt signaling, beta catenin signaling. So we just screen all of the factors involved in the Wnt signaling, and we use the quantitative PCR and test the beta catenin and TCF4 transcription factors and all the others. And we find the high-fi diet inhibited in the transcriptional expressions of beta catenin, TCF4, and axin-1 in cortex, no matter is under or without the cadmium exposures. So for the early life cadmium exposure, did not affect the transcriptions of those Wnt signaling components. But when we run the proteins, we can look at here, compare the different dose, 0.5 ppm, 5 ppm, we can see that's more active beta catenin accumulate in the cortex. And compare only in the exposed to the high-fi diet, we can see these active beta catenin accumulated a lot in the tissue, and the one of the factors is GSK3 beta, phosphorylated GS3 beta is increased. But under the 0.5 ppm and 5 ppm, we combine these high-fi exposures. It's not that significant and difference. We cannot see that. So this is raise the question, can early life cadmium and or high-fi diet exposure cause the epigenomic alterations in offspring mouse brain? Because the transcription factors not change, but protein level is changed, so we consider that. And this is we run a pilot test. We just look at that DNMT1 epigenetic enzymes, and we find some difference at transcriptional levels. So here is the conclusions. So the first early life exposure to 5 ppm cadmium in drinking water is the marginal dose for behavior impairments and pathological changes in mice. So in the future, when I decided when I submit the grant, I set up another group is give a little bit the double dose for the cadmium. And second, the mice fed cadmium rather than high-fat diet contributes the ASD relevant behaviors. Mice fed high-fat diet may cause anxiety-like behavior. And the fourth conclusion, early life low-dose cadmium exposure or post-weaning high-fat diet cause DNA oxidative stress, glia activation, inflammatory response, and BBB impairment in offspring brains. And post-weaning high-fat diet exacerbates the cadmium-induced DNA oxidation, glia inflammatory response, and BBB disruption. The Wnt-based catenin signaling is a candidate we would like to look at further. So here is the lab. Wendy is a technician and contribute most of the behavior test and real-time PCR. And Teresa is around the immunofluorescence study, and she's now moved to Cincinnati and get a better job position. Yeah. And also, I would like to thank you for Dr. Stace for that pilot grant and let me have a chance to step in a new field.